Antiretroviral therapy is one of the drugs used to treat HIV and AIDS. It is not a cure, but it's a treatment. What it does is it targets the HIV cell replication. That's an HIV cell. HIV cell replication is a multi-step process. It has a lot of steps. The first one is the HIV cell is going to find a host, or a T cell. T cells are part of your immune system. The HIV cell is going to attach itself and pretty much turn the T cell into an HIV cell factory. The T cell has receptors. Those are the yellow things. Receptors are going to work kind of like our mouths. We only eat things that are good. We're not going to eat nasty stuff like rocks and dirt. Much like the T cell is not going to take in things that are nasty and bad for it. But the HIV cell has the little orange things are proteins, which are good for the cell. The T cell is going to try and take in the proteins of the glowing things and, trying to absorb them and use them. So the HIV cell is going to come towards the T cell, and it's going to attach the orange protein things to the receptors. The receptors are going to attach to the orange things. This is actually called attachment. When this happens, when they're finally attached, the blue things, the attached the HIV cell to the proteins, are going to unfold. They don't actually disappear in real life, but just so you can see it better. They're going to unfold and they're going to attach the HIV cell to the T cell. Then they're going to fold and squish and, compress to pull the HIV cell and the T cell together. The HIV cell's membrane is going to fuse. It's going to become part of the T cell membrane. It's going to dissolve into the T cell. Inside the HIV cell are 3 parts. The 2 strands of RNA and 3 replication enzymes. The replication enzymes are going to be like the workers of the HIV cell factory, which is what the HIV cell is going to turn the T cell into. The T cell is going to become a factory, and the 3 replication enzymes are going to be the workers, and I guess the RNA is going to be kind of like the supplies to make the big computer, I guess? I don't know. Anyway, so the 3 enzymes are the integrase, the protease, and the reverse transcriptase. There's 2 parts to the reverse transcriptase. I can't say either of them. But what the reverse transcriptase does is it's going to mess with the 2 strands of RNA and make it into something else. Okay, so here we go. The first part of the reverse transcriptase is taking the 1 strand of RNA and turning it into an RNA. The RNA is the red one. RNA DNA, the DNA is the blue one. RNA DNA double helix. Then the second part is going to split the RNA DNA helix apart, and then the DNA is going to go back around to the first part, and it's going to become a DNA double helix. So there's 2 strands of DNA now. So the first part, it adds DNA to whatever. That is integrase. The integrase is going to chop off the 2 ends of the DNA double helix to make it like sticky. Like you would put glue on something to attach it, the integrase is cutting those off to make it sticky so it can attach it into the T cell's DNA. So the integrase is integrating the HIV double helix into the T cell's DNA. The HIV DNA is going to produce a RNA messenger, which is going to then travel into the cytoplasm, which is the outer layer of the cell. That RNA is going to create the building blocks for the new HIV cell, which are those long purple protein strands. Then the protease is going to cut some of the long protein strands into smaller protein strands, which are going to become the basic building blocks of the cell. This is essential for making a contagious virus. Then, 2 RNA strands, all of the factory workers and the proteins are going to assemble they're going to leave the cell, and as they're leaving the cell, they're going to take some of the T cell's membrane, which has got all the good protein stuff that the HIV needs to disguise itself. Then it's going to mature and become an HIV cell and go infect other T cells. This happens billions of times per day. This is very bad. It kills all of your T cells and completely ruins your immune system, leaving you prone to many diseases, which is what will eventually kill you. Antiretroviral therapy drugs are to stop. Our goal is to stop the reproduction of the HIV cell, which destroys the T cells. The first thing that happened was the attachment. This is going to be stopped by... The attachment process is stopped by fusion inhibitors. That's that gray thing. It's going to come around and stop the blue things from unfolding. The second thing that happens is nucleoside inhibitors are going to stop the reverse transcriptase, one of the workers, from turning that RNA into the RNA-DNA helix. This is the common standard drug used in many antiretroviral therapy. The next thing it's going to do is it's going to stop the integrase from putting the HIV DNA into the T cell DNA. This is also very common. The next thing it's going to do is it's going to stop the protephase from making the essential compact proteins. This is also a common drug in many antiretroviral therapies. The goal of the antiretroviral therapy drugs is to slow down the process of the HIV replication. I didn't make this video, the Ingeheim, whatever institute made it. So, thank you for watching.